Well, good morning, good afternoon, or good evening, ladies and gentlemen. Welcome to another installment of our video PowerPoint series for Unit 5, Cell Energistics. Today, what we're going to be talking about are cell membranes and transport. So we're going to be talking about what the cell membrane is, what it does, and how it allows certain things in and out. What we're also going to be discussing are things like osmosis, diffusion, and facilitated diffusion and active transport. So primarily all we're going to be talking about today is how things go in and out of the cell. So, without further ado, let's go ahead and get this thing started, shall we? I'll see you on the next slide. So homeostasis. Now we talked about this quite some time ago, but homeostasis, if you remember, is the balance that cells must maintain with their surrounding environment. So homeostasis, it's all about that balance, equilibrium. That's what all cells want. Now, cells maintain homeostasis by controlling what enters and exits the cell, because as we know, the cell membrane goes ahead and is much like a bouncer, controls what comes in and what goes out. So the cell membrane, it's made of a bilayer of phospholipids. So phospholipids, we're talking about lipids primarily, so fats, things that don't normally mix that well with water. So phospholipids have a hydrophobic tail, which is water-fearing, and a hydrophilic head, which is water-loving. We also have certain types of proteins that are embedded in the membrane to help materials move in and out of the cell. So if you all go ahead and take a look down here, so we have our hydrophilic head, hydrophilic meaning it loves water, and we have our hydrophobic tail. So if you take a look at this, the heads are all facing outwards, either into the extracellular fluid or the intracellular fluid, whereas the tails, which are hydrophobic, are facing each other, making kind of a water-sealed barrier between the inside and outside of the cell. So the membrane is called a fluid mosaic. So based off what we saw in the last slide, why do you think that's the case? Go ahead, take some time. Think it over. So, now granted I can't actually hear what you're saying, but I'm assuming what you came to was if you look back on the last slide, you saw that you had all these tiny little phospholipids embedded in a fluid. So a fluid mosaic, if you look at these pictures here, the mosaics are all made of tiny little pieces that are put together in a certain way to make a picture. So same thing with the cell membrane. The cell membrane is a bunch of tiny little parts all embedded in a fluid, hence why they call it the fluid mosaic. So here we have the cell membrane. So if you'd be so kind, I'd like you to go ahead and take a quick sketch of this and draw this in your notebook. Now, you don't have to go ahead and make it all artistic, make it all detailed and three-dimensional, but what you're going to need is you're going to need the outside of the cell, you're going to need the cell membrane itself, and the inside of the cell or the cytoplasm. So there are some certain aspects of our cell membrane you need to include. So if you look, of course, well, you need the lipid bilayer, which are our phospholipids with the hydrophilic heads and the hydrophobic tails, facing inwards towards each other. What we also have are the protein channels. So the protein channels, which we'll talk about in a little bit, the protein channels allow larger material and molecules to actually transfer through the cell membrane without diffusing through. And we also have the carbohydrate chains up here. So the carbohydrate chains kind of act like little signals. They stand there waiting, and once they come in contact with something that can be pulled through the cell, they send out a signal telling it, hey, we can bring this in. So, go ahead, pause it if you need to, but I'd like you to draw this down. Well, I'll see you all in the next slide. So the cell membrane has control over what can cross it, so that only certain molecules can either enter or leave. Now this is what we call selective permeability. So selective permeability, think about this for a second, controls what goes in and what goes out. Are there any household appliances that may or may not do the same thing? Give it some time. If you've ever made things like macaroni and cheese or pasta, you might use a strainer or colander. So very similar to a colander or a strainer that lets water pass through but it keeps the pasta in, it's selectively permeable. So much like the cell membrane, it determines what comes in and what goes out. It lets certain things through, but not others. So the first thing that we're going to be talking about when it comes to transport is passive transport. So passive. Passive is very relaxed, very chill, and it's not going to require any energy. So it's the movement of molecules across the cell membrane that does not use energy up. And materials will be flowing from an area of high concentration to an area of low concentration. So think of it like this. You're going ahead and floating downstream with a river. So since you're in the current, you don't really have to expend any energy to go downstream, to move. Very similar to this. So with passive transport, as you can see, we have a high amount on this side and a low amount on this side. So over time, what happened here is that our solutes, our molecules, move from one side to the other until we have balance. And like we said before, all cells are looking for that homeostasis, that balance. So diffusion. Diffusion is the movement of molecules through a cell membrane. It's a type of passive transport that does not use any energy. So what we were talking about in the last slide is basically diffusion. So think about it when you go ahead and put something like food coloring. When you put a drop of food coloring into a cup of water or a beaker of water, the color is very, very dark initially, right? But over time, it's going to go ahead and spread throughout the rest of the beaker or the cup until you have one big uniform color. So based off what we talked about here, where do you think the molecules are going to move? Are they going to move from left to right, A to B, or right to left, B to A? If you said they move from left to right, you better believe it. That's correct. Now this was the case because, like we said before, there are more molecules on the left-hand side with A than there are on B. So over time, they're gradually going to move until we have equilibrium. So when is equilibrium reached? Equilibrium is reached simply when the concentrations are equal on both sides of the cell membrane. We have that balance. So osmosis. Now osmosis is just like diffusion, however it's a specific type. It's the diffusion of water molecules across the cell membrane. Now since it's diffusion, once again it's still passive transport. No energy is being used. Now water always diffuses from an area of low solute concentration to an area of high solute concentration. So what that means is, think about it like this. We know that diffusion goes from high to low, right? So that means if we have a low solute concentration, then we have a high amount of water, or H2O. Sorry, I'm using the trackpad on this. It's kind of sketchy. And likewise, if it has an area of high solute concentration, that means it has a low amount of water. You know, for a trackpad, that's not bad. Anyway. So the way that I always look at this is it's always going to flow from an area of high water concentration to an area of low water concentration. High to low. And by the way, just a quick little reminder. Do you guys remember what a solute is? That's right, a solute is what's dissolved inside of a liquid. So it's kind of like Kool-Aid powder. So just like we talked about before, as you can see, on this left-hand side here, we have way more solute than we do solvent. So we have way more solute than water on the left. And we have way more water on the right compared to the two ratios. We want them to be equal. So over time, what's going to happen here is the water will move from the right side to the left side to balance out the two equations. We want things to be equal. We want an equal amount of water to solute ratio on each side. So osmosis, once again, is driven by solute concentration. So the first type, specifically, that we're going to talk about is something called hypotonic. So in a hypotonic solution, the solute concentration is lower on the outside of the cell than the inside. So what that means is we have more water on the outside than we do the inside. Now to balance this out, the water is going to go ahead and rush inside the cell. Now when you have all this water rushing inside the cell, it's going to swell up. So water is going to be moving into the cell, causing it to swell up and get a tad bit roly-poly. Now, the way I remember this one is hypo sounds a lot like hippo. And both are kind of rotund, right? So hypotonic, water rushes in and causes the cell to swell up. On the other end of the spectrum, we have hypertonic. So a hypertonic solution is where the solute concentration is higher on the outside of the cell than the inside. So what that means is that we have more water on the inside of the cell than we do the outside. So what's going to happen is the water is going to rush out of the cell, causing the cell to go ahead and shrivel up. So hyper means excessive. So the way that I always remember this one is, think about it. Okay, you're babysitting a kid, all right? You're babysitting a child. And the kid is just going nuts. The kid's bouncing off the walls. So what do? You go ahead and you take the kid outside the cell. You take them outside the house, you let them run around for a bit, do whatever they need to, and then they're good, right? So, hyper, water leaves the cell shrivels up. The final one that we're going to talk about is isotonic. In an isotonic solution, the solute concentration is equal on both sides of the membrane. So water diffuses in and out equally. So we have a balance on both sides. Now this doesn't mean that they just stand still. That means that water is going in and out of the cell at an equal ratio, so the cell stays about the same size. No worries. Iso, once again, means equal. So osmosis and plant cells. Plant cells require a hypotonic environment, so water moves into the cell and the cell swells up. The swelling of the cell builds up turgor pressure. So think of it like this. When you don't water a plant for a while, what happens? It starts to kind of wilt, right? It starts to shrivel up. So if you go ahead and water the plant regularly, you're keeping the vacuoles, that's right, I went back to the cells unit, you're keeping the vacuoles filled up with water. And when that happens, that plant's going to stand right up. It's going to stand up straight, and that's what makes it happy. So plant cells in a hypertonic environment lose water and the cell starts to shrivel up. The shriveling of the cell is what we call plasmolysis. Plants don't like this. Now, for example, osmosis. Let's think here for a sec. In marine fish. So we have two types of fish, right? We have saltwater fish and we have freshwater fish. If you take a freshwater fish and put it into salt water, that means that we have more water on the inside of the fish than we do the outside. So what's going to happen here is the water is going to leave the cells of the fish, causing it to actually die from dehydration. And on the other end of the cell, or at the other end of the spectrum here, if you took a saltwater fish and put it into a freshwater pond, what would happen is there's more water on the outside of the cell than the inside. So the water would rush into the cells of the fish, causing them to go ahead and expand and, well, kill off the fish. Now this does not mean that if you go ahead and toss a narwhal into a lake, the narwhal is not going to explode, okay? It's not going to be like, bye, buddy, hope you find your... No, that's not how it's going to work. Sorry. That's why you always want to keep saltwater fish in saltwater and freshwater fish in freshwater. So let's think back here. An animal cell in hypertonic solution. What do you think is going to happen to it? It's going to shrivel up. Hypertonic, water moves out. In an isotonic solution, however, animal cells like this, so they stay relatively balanced. In a hypotonic solution, water is going to be rushing into the cell, causing it to swell up, and if it doesn't stop, it will eventually explode. In a plant cell, in a hypertonic solution, what happens is once again the water leaves the cell, causing it to go through plasmolysis, or it shrivels up. In an isotonic solution, that means the plant cell is going to be flaccid. I mean, it's going to have some water in it, but not nearly as much as it needs. So it's going to be kind of rubbery. And in the meantime, if we go ahead and put it in a hypotonic solution, it's going to be turgid. It's going to be filled with water. It's going to be happy. That's what the plant wants. So facilitated diffusion. Once again, this is still a type of diffusion, so there's no energy being used. What this allows to happen, though, is molecules move in and out of the cell using a carrier protein. So a carrier protein, why would something need to use a carrier protein to get inside of the cell? Well, the reasons why is that it might be too big to normally diffuse through the cell membrane, or it might have a charge to it. If it has a charge or it's too big, it's not going to be able to make its way through the cell membrane on its own. So what's going to happen is it's going to go ahead and be brought in through the carrier protein. So active transport. We talked about passive, where no energy was being used. So active transport is just the other type. So active transport is the movement of molecules across the cell membrane that requires energy. So this can move materials from an area of low concentration to an area of high concentration. So you're going opposite of diffusion. So think of it like this. If you're going ahead, if diffusion is like sitting in an inner tube or just kind of floating there and going downstream with the current, if you want to swim upstream against the concentration gradient, against the current, you're going to have to use some energy. The same thing with active transport. So in this case, we could actually go from right to left. We could go from the low concentration to the high concentration. So why does your body need to use active transport? Now this is all up here. Up inside your brain, you have neurons. And for these neurons to go ahead and transfer signals from one to the other, it uses a series of sodium and potassium pumps, which go ahead and utilize active transport to carry the concentrations over. Now, if you'd like to, please go ahead and watch the two animations below in the video. It's going to help you understand this. Now, I'm not going to go ahead and watch these with you because, well, I can't. So I'll be here when you get back. Go ahead, give them a look, see what you think. Alright then. So, the last two things that we're going to discuss here with active transport are endocytosis and exocytosis. So, endocytosis is where large molecules can be engulfed by the cell. So we're bringing things inside the cell. Now we have two different types. We have penocytosis, which is the process of engulfing a liquid, or cell drinking. We also have phagocytosis, which is the process of engulfing a solid, or cell eating. So as you can see, what happens is the molecules go ahead and start to push on the cytoplasm. Now it keeps pushing and keeps pushing and keeps pushing until it eventually creates its own storage molecule, kind of like a box. And these boxes are what we call vesicles. So it goes ahead and keeps pushing on the cell membrane until it creates its own separate vesicle. So we're bringing substances into the cell via active transport. The second type is exocytosis. So if endo was to bring in, exo is releasing material from the cell. So same thing is with endocytosis, but in reverse. So the vesicle is going to go ahead, join in with the cell membrane, and actually release. So the vesicles, the phospholipids will become part of the cell membrane once again, and then they will release their contents out into the extracellular fluid. So endo means bringing in, exo means releasing. So with that, ladies and gentlemen, that brings this lesson to a close. Thank you for joining us for the first video of Unit 5, Cell Energistics. So today we talked about diffusion, osmosis, passive transport, active transport, and the cell membrane, as well as what the cell membrane does. So in retrospect, the cell membrane is made of phospholipids. It determines what comes in and what can leave the cell. So it's very selective. It determines what goes in, what goes out, very similar to a strainer making pasta. Lets the water out, keeps the pasta in. We also talked about passive transport. So passive transport is the movement of material across the cell membrane without requiring any energy. So it goes from a high concentration to an area of low concentration. So with this, what we were discussing was diffusion. And specifically, we also talked about osmosis. So diffusion is just the movement of molecules across the cell membrane. Osmosis is the movement of water. So with osmosis, we talked about three different types. We had hypotonic, where there's more water on the outside of the cell than the inside, causing water to rush in and make the cell swell up. There's also hypertonic. So hypertonic, we have more water on the inside than we do the outside. So the water is going to leave the cell, causing it to shrivel up. We also talked about isotonic. So in an isotonic solution, we have an equal amount of water on the outside and the inside. What this allows the cell to do is we have an equal amount going in and out. So the cell's happy. It's not going to increase or decrease in size, it's going to stay stable because it's reached homeostasis. Now, we also talked about one more type of passive transport, which was facilitated diffusion. Facilitated diffusion allows larger molecules that are either too big, can't diffuse through, or have a charge to pass through the cell membrane with the use of carrier proteins. Now, we also discussed active transport. So active transport is like swimming against the current of a river. If you can still move that way, but you're going to have to spend some energy to do it. So with active transport, we're going against the concentration gradient. We can move from low to high. Now, we had two different types that we talked about. So the two types that we talked about were endocytosis and exocytosis. So endocytosis is the process of bringing materials inside the cell. The two types we talked about were phagocytosis, which is cell eating, bringing solids in, and penocytosis, which is bringing in a liquid. And we also talked about exocytosis. So exocytosis is removing material from the cell. Well, that's just about going to go ahead and do it for us today. I'd like to thank you all for going ahead and joining us on this fine day. And until next time, I will see you in the next video. You all keep it classy.